Hi, everyone. Good afternoon. My name is Fan Liu. I'm the co-founder and CEO of neoX Biotech. We were founded in 2018, and we were the first AI-driven biotech in China that designed biologics and this bispecific new wave drug modality. We currently have 24 people, and we are located mainly in Beijing, Shanghai, and Boston. So what we really focus is so-called protein-protein interaction. Basically, this complex move of how one protein recognizes the other protein and carrying out its drug function. And we focus in the therapeutic area of oncology and immuno-oncology. Our business model is develop the drug from scratch to a certain level before entering into human, and then sell the right of the drug to multinational pharmaceutical companies that would carry on the drug development in clinical phase. So we specialize in the early discovery of new drug modalities. And this kind of endeavor really requires a fusion of different expertise, both computational and experimental. So I was trained as a computational chemist at Caltech, and our SVP of oncology, Dr. Yang Xie, was trained as a biologist from Yale, and then later worked in Novartis and several innovative biotech, biopharma. And really the core team is this combination, this integration of AI scientists from ByteDance or drug hunters from the pharmaceutical companies. And the close collaboration is really the key in this new wave of AI-driven drug design. We work in two categories of drugs. On the left, biologics. On the right, degraders. And there are these new waves of biologics, such as nanobody, which is only half the size of traditional antibody, or even smaller, such as mini protein, which is only 1/3 of nanobody, and even smaller, like peptides, cyclic peptides. All this new scaffold of biologics requires, there is lack of empirical rules, but requires new design principles coming out of data, out of first principle calculation. And on the right, there's these bispecific degraders, which I will delve into later. And both of these two categories of drugs are based on the protein-protein interaction, not the traditional small molecule where just a small molecule binds into one target. So that picture does not apply to these new waves of drugs. And this is just a taste of how complex this protein-protein interaction problem is. So usually in X-ray structure, you take a static picture of the protein-protein complex, or AlphaFold algorithms, it predicts one static picture of how protein recognize each other. But indeed, its process is quite dynamic. If we perform this simulation up to microseconds, you would observe an ensemble of conformations. And in delving into statistical analysis, you would see in the middle this free energy landscape. Basically, this is the map of the function of protein-protein recognition. With this map, we can then identify the conformation, the structure on the right, which you cannot obtain experimentally, but only from computer simulation. Once we have secret look into this transient but functionally relevant important conformation, we can then design mutations to fine-tune the property of this protein to achieve our desired property. And also, new algorithms are needed to help improve the drug property. For example, biologics, we want to solve the problem to predict the so-called immunogenicity accurately. High immunogenicity makes the drug not effective. Our body would generate antibody that get rid of these drugs and making it not function at all. So we are the first to use transformer, the famous T in ChatGPT, into developing this algorithm and obtained the best of the class property precision of this kind of algorithm. And the application scenario would be given a new biologics target, a new biologics sequence candidate. We would use this algorithm to scan the sequence of that biologics and finding all these weak spots, all these liabilities showing orange. And in the drug screening process, we want to in silico get rid of, minimize all these orange spots. Let me use an example. So this protein is called interleukin 21. This is one specific type of cytokine which can be used to further stimulate the immune cells inside the tumor to activate our immune cell, our own defense system to recognize the tumor and kill the tumor. However, there are two liabilities of the natural format of this protein, of this interleukin 21. Number one, it's not stable. It has a really short half-life, and you don't want the drug to be that transient. Secondly, it binds to its receptor really tightly. So once it binds to an immune cell, it does not like to get off. And that means once you inject that natural format into the blood, it just stays in the immune cell, in the blood or normal tissue, not hopping into the tumor inside the tumor-penetrated immune cell. That's the exact properties we want to engineer. And we perform this kind of conformational analysis up to its key dissociation and association process and find these 4 very important key sites which controls that dynamics, that kinetics of the protein. And we use AI algorithms to generate 400,000 sequences. Those are completely de novo computer AI-generated candidates. And using the set of algorithms we developed in-house, we further score its different properties, such as affinity, stability, and immunogenicity. So that computational work takes 1 week, and the experimental testing takes 1 month. So basically, in 1 month, instead of 6 months' time of the traditional method, we got the first batch of feedback. And that requires further optimization for a second round. And in 2 months, 2 rounds of iteration, we got our drug candidate, which has the following property, where in the left, it shows that the later curve, it binds fast, it dissociates fast, it can hop faster into the tumor. In the middle, it shows that the denatured temperature of this protein with our designed for mutations improved significantly. It becomes more stable protein. And all these 2 properties combined together, on the right, you see that in this animal, so-called mice, tumor-bearing mice model, it really kills the tumor, inhibits the tumor to 60–70%. And that forms basically a good drug with all the designs that we put into the protein. And another pioneering pipeline for neoX biotech is this anti-CD24 monoclonal antibody. This is a very new target that's highly expressed in ovarian tumor, in breast cancer, in lung cancer. However, with all the desired property of that monoclonal antibody, the true challenge comes, how do we design an antibody that is tumor selective? And for this target on the right, the picture is showing that green peptide is the target. There's a special tumor marker, special patch of area for the target on the tumor that is different from that target in normal cells. And we really want to design an antibody that recognizes that specific batch of area, so-called epitope. And we fine-tuned and improved AlphaFold2 so that now it can high confidently predict where does that antibody binds. And with that screening process of that algorithm, we were successfully finding the unique, the only tumor-selective anti-CD24 antibody, where we recently get US FDA IND approval. It is ready to put into human. And we have this comprehensive data set with cell tumor killing data, with the animal tumor-bearing mice data. So this forms a complete package where we can license out the global right or out of China right to multinational pharmaceutical companies with decent amount of upfront payment to further upgrade our algorithm, upgrade our platform, and innovate the next drug. So let's switch gears to the degrader space. Degrader is also a complicated drug. It engages two proteins. One is the target. The other is the enzyme that modifies the target, make the drug degrade. And the traditional picture is on the left. People use these simple algorithms to design one specific protein conformation, but our innovation method basically considers a group, a set of, or so-called ensemble of conformations that all contributes to the stability of the desired protein-protein complex. And with that, we can accelerate the so-called degrader design with the following iterative loop, where we design, again, we generated the candidate molecule, we score it in this complex protein-protein complex formation, and in each round, we nominated 10 to 20 molecules to synthesize, and then test and iteratively improve. With 3 rounds of iteration, and with only less than 100 molecules synthesized in material, and with less than 1 year, we can nominate our preclinical candidate, comparing to the industry standard reported, which is 3 years and 400 molecules synthesized. And our drug candidates has been accepted in top 2 oncology conferences and invited talks. We really want to upgrade the degrader game. So first of all, we design only the linker part of the molecule. And second level, we upgrade the game to design the whole molecule. And the third level, we really want to rationally design a kind of degrader called molecular glue. Traditionally, molecular glue is identified by serendipity, meaning luck, doing this kind of screening wet lab process. But with the algorithm that we developed, we can now computationally identify putative target for molecular glue through this process. And once we identified a target from the first principle, and then we virtually, in silico, screen the hit molecule, the first molecule that binds to the target in this context of complex protein-protein interface, and can really degrade that target, which no previous molecules could do. And the key essence is, again, in the middle, this ensemble of conformation, this so-called high entropy context where the current algorithm could not solve. And with that flexible pocket getting involved in that algorithm, we were successfully identified virtual hits and then tested in experiments. So these circled 7 hits are validated experimental hits that can form a stable protein complex. And in the middle, one of the 7 hits can actually carry out the function, degrade the target, making this band look like the diner in terms of the color. So, in the past 6 years, with our protein-protein complex-based AI algorithm, tackling this highly complicated protein complex conformational problem in 2 categories of drugs, one biologics, showing this Y antibody-like label, and the other degrader, which is this kind of new format of bispecific molecule, we were able to generate the following pipelines from our platform. And we are ready and in active business partnership with potential partners, getting good upfront payment and with good algorithm co-discovery projects. And we are luckily funded by some famous venture capitals in the past 6 years, really believing in that AI can accelerate the drug process, acceleration process. If you'd like to get to know more about our platform, know more about our business model, more about our innovative drugs, feel free to shoot me an email. Thank you.